Practice 4. Nucleic acid extraction. To study the molecular genetics of organisms, it is necessary to extract the good quality DNA, using different techniques such as PCR, molecular markers, sequencing. To choose the right method, it is necessary to know the nature of the cell, from which it must remove and thus be able to purify in the future. The insulation essentially consists on cell lysis, protein removal, precipitation for concentrating nucleic acid. The most important considerations to extract genetic material are obtained from samples. The cell wall is degraded with enzymes and the membrane with detergents. Removal of proteins is performed with phenol, chloroform, and isoamyl alcohol, and the precipitation is with ethanol or isopropanol.
For the realization of the practice was needed an extraction of genomic DNA from human blood and an extraction from total ARN from Escherichia coli, strain DH5 alpha. In the case of the human blood, wasn't necessary to do something before the practice, but in the case of the ARN from bacteria, was needed to do a culture with the bacteria a day before, in Luria bertani broth. Then, the extraction, by the optimization of time, was scheduled to do all the process in 2 sessions. In the first one, was done the half procedure of the extraction of DNA, and almost all the procedure of the extraction of ARN. In the second session, was obtained totally all the extraction of DNA and RNA, and was time to prepare the electrophoresis in a Yaros gel. The electrophoresis was totally done in the session with 120 volts. In the electrophoresis wells were incorporated a mixture from 990 microliters distilled water and 10 microliters with the water molecular biology grade with ARN and other with DNA, and also the DNA ladder. In this session was used the 1 kb plus. The other wells were used to the practice. In addition to the electrophoresis gel, was used a spectrophotometry for determine the concentration of DNA or RNA in the results, using a factor 1 absorbance in 260 or 40 micrograms per milliliter.
Results. Using an spectrophotometer, the purity of the sample can be obtained. This gives you an idea of how much DNA or RNA is really present in your sample. The absorbance index was measured and obtained. The next value. For DNA it was 1.7 and for RNA was 1.68. Afterwards, the procedure for the electrophoresis gel was carried out, and the results of the bands were visualized. There were 2 bands for DNA, one darker than the other, and for RNA there were several bands, but more defined ones. Discussion. The ideal index 260-280 for DNA is 1.8, and for RNA is 2. This means that the results obtained previously weren't in the desired levels. Nonetheless, the bands were visible. However, this also may mean something more, such as contamination. When making a gel, it's advisable to give the sample a pretreatment to avoid this type of contamination with other molecules such as proteins or a determined nucleic acid such as DNA or RNA. Therefore, it is advisable to pretreat the sample with enzymes that can degrade such molecules. This is for that the results obtained in the electrophoresis gel showed precisely this, the contamination with proteins and RNA and DNA. It is advised to use an enzyme that can degrade protein or make another phenolic extraction, just as well for the other as nucleic acids.
As conclusion, it's necessary to consider many aspects at times to isolate genetic material of a different nature, since, as we'll see, there are cases in which the running of this material in PCR has contamination of protein, RNA, or DNA unwanted. To avoid this contamination, must follow specific protocols for each material in order to achieve the material purification, and then get more accurate and specific results. This protocol should include optimal amounts of compounds such as enzyme or detergents, and among others, that are required for each sample, and also include the time of washing, drying, spin, and even electrophoresis to appreciate a better binding and counting of the obtained samples. Finally, it is important to know the ways in which it can be seen if a PCR genetic material is contaminated or has a problem when you are viewing it using ultraviolet light.